discovery in the field in the last couple of years was the discovery of so-called cascade. And cascade is a complex of 6 Cas proteins that performs the key function in the CRISPR system mediated defense. This cascade complex has a very, very peculiar, beautiful structure with a great resemblance to a seahorse here. Quite an interesting, rather asymmetrical structure. Typically, molecular complexes that you see are very symmetrical, with a few prominent exceptions like the ribosome. And this cascade is yet another prominent exception from this symmetrical pattern, and it has been shown in several papers by now, the structural organization of this complex has been characterized in great detail, and it has been shown which of the Cas proteins forms the structural scaffold, found in 6 copies here, and which ones, this one in particular, is the active nuclease moiety, and it has been shown that this gate complex positions the CRISPR RNA in a unique manner here and is responsible both for processing of the primary transcript into the small guide RNAs and to a large extent for the interference itself. So we recently analyzed in some detail and summarized in this paper the evolution of what we can understand about the evolution of CRISPR-Cas systems and the new classification that was developed and that I will briefly describe in a moment. But before I do that, let's just review the schematic of the functioning of this system as we understand it now. So there are 3 different types of functions, somewhat differently, but the basic steps are the same. So in a poorly characterized series, in the series of reactions that is still poorly characterized, two of the Cas proteins, apparently probably the proteins denoted Cas1 and Cas2, about which we will talk a little later, recognize a certain nucleotide motif in alien DNA. They recognize the so-called PAD motif. They manage somehow in a manner that is not characterized yet, excise a piece of DNA and integrate it into that piece of alien DNA that is called proper spacer into the CRISPR repeat cassette. And then this creates a potential for immunity, which is realized by expression of a long pre-cRNA transcript that is processed by cascade complex. The structure of the complex differs somewhat between different types of CRISPR systems, and then the same protein complex bound to the guide RNA and with the help of an additional Cas protein mediates interference that actually includes, at least apparently in most of these systems, cleavage of the bacteriophage or plasmid DNA itself rather than silencing of phage RNA as we proposed initially based on the analogy with the eukaryotic RNAi system. That question is still not solved, and there are indications that RNA may be recognized by some of these systems, but apparently the primary mode is degradation of the foreign DNA itself, and of course it makes a lot of sense, if you will, for a prokaryotic immune system to function in just that manner because, well, bacteria and archaea are awash in all kinds of forms of DNA, phage, plasmids, and whatnot, and this system provides powerful defense, along with other defense systems such as restriction modification. So by comparative genomic analysis, we developed this classification of the CRISPR-Cas systems into three distinct types, each of which has its own signature. Genes that are boxed in this figure, you don't have to go into the fine details of all these gene organizations, but just visually you can see that these systems are, while sharing some components, they're extremely diverse and extremely different and show highly diverse gene organizations. We also naturally did phylogenetic analysis using the Cas1 sequence, the most conserved sequence, the ubiquitous and most conserved sequence in the CRISPR-Cas system, and take the tree like this. I've shown you just as a schematic. It has all the original tree has all the bootstrap support and all that kind of analysis, which shows interesting things. It shows that mostly these different types and subtypes of the CRISPR-Cas system are phylogenetically coherent. In particular, this applies to type 1 and type 2, but type 3 is in a certain sense different because it's split into several branches in the tree, like here, here, here. So along with some phylogenetic coherence, we seem to detect considerable evidence of rearrangement and recombination between these gene loci. Then analyzed in detail the relationship between these ramp proteins. Remember, I introduced that in the very beginning of my talk as repair-associated mysterious proteins. Now, they have been recrystallized into repeat-associated mysterious proteins, say with the acronym, and they have become somewhat less mysterious, thanks in large part to the determination of several crystal structures, which all revealed a particular kind of domain, the so-called RNA recognition motif, which is a variety, which is a subtype of the so-called ferredoxin fold, a very basic beta sheet fold. And there are three distinct groups of these proteins, some of which appear to contain a catalytically active histidine, which is denoted by this yellow bar here, and apparently are active nucleases, and our reconstruction suggests that this was the ancestral form, a single RRM domain with an catalytically active histidine, an active nuclease. And then these proteins really have undergone a lot of evolution, such as loss of, such as duplication of the RRM domain, deterioration of, disruption of RRM domains, loss of these domains, loss of the catalytically active site, and so on and so on and so forth. This classification looks neat, but if you start comparing these sequences using BLAST, for instance, you'll see very little. They are extremely highly diverse proteins. It requires a lot of effort and other subtle analysis to come up with this classification. These genes encoding these RAM proteins show very distinct arrangements in different subtypes of the CRISPR system of Cas operons. And what I want to emphasize here is that actually all these extremely diverse prokaryotic immune systems are really centered around one single domain, the RNA recognition motif here, which I have shown here as a very typical structure that contains a simple anti-parallel sheet of 4 beta strands and with some embellishments that vary, and that binds an RNA molecule which pegs against the surface of that beta sheet, a very basic domain that importantly is found in a huge variety of proteins, not only not at all only in these RAM proteins, but in much better characterized proteins, such as various kinds of polymerases, for instance. And indeed, within the CRISPR-Cas system itself, it's not only these RAMs, Cas5, Cas7, Cas6, that contain an RRM domain, but completely different proteins as well. For instance, Cas2, which is involved apparently in adaptation, spacer, acquisition, as well as Cas10, which hasn't been biochemically characterized yet, but is predicted in some of these systems to be an active polymerase. So it's quite amazing that this extremely complex system is almost all generated by the diversification of a very basic, simple RNA binding domain. So we came up with a speculative, of course, but a relatively simple and I think parsimonious scenario for the evolution of this system, which based obviously on a lot of comparative genomic analysis, which suggests that the system really came together from 2 distinct components. One of which is the cascade complex, which is based almost entirely on RRM recognition motif domains, RRM domains, and might have evolved from other better known RNA binding proteins such as polymerases, or maybe so-called bound domain of polymerases, which is also an RRM domain, but possibly also from the recruitment of other RNA binding proteins, because there are many. And we hypothesize that in the beginning of the evolution of this system, this was a standalone complex that might have been functioning by generating guide RNAs that inhibit reproduction of alien DNA, but not integrating it into the host genome. And then it got combined with an element of different origin, consisting of Cas1 and Cas2 genes, which provide for the integration stage. So this was about, so to speak, macroevolution and origin of this system. They then decided to study the microevolution of this system. And one of the best ways to study microevolution is to assess the power of selection that affects genes. For that reason, you need groups of closely related genomes, and if you have such groups, you can rather accurately estimate the so-called dn over ds ratio, which is the ratio of non-synonymous mutations that change protein sequence to synonymous mutations, and the lower this ratio, the stronger the refined selection affecting given genes, somewhat predictably, but in a satisfying manner. They found that compared to other genes in their respective groups of genomes, the Cas genes are subject to very weak purifying selection.